This video goes through the formation of tissue fluid from the transport in animals topic of exchange and transport. Two key terms referenced in the spec on the right side are hydrostatic pressure and oncotic pressure. OCR favour these terms, however other terms are often found on diagrams, including the ones I have used. A blank version of this worksheet should be found in your classwork folder, and the completed version that you can see here is in the content library in the transport in animals folder. Tissue fluid is formed from the capillaries at the arterial end. This is because the blood flows into the capillaries of the tissues at a high pressure from the arteries and arterioles. This high blood pressure, hydrostatic pressure, forces small molecules out of the blood plasma through small holes between the epithelial cells of the capillaries. This includes molecules such as dissolved gases, dissolved minerals, and also dissolved nutrients. Other contents of the blood, such as red blood cells, white blood cells, platelets, and plasma proteins, they're all far too large to fit out of the capillaries by ultrafiltration. They're far too large to fit through the small pores between the epithelial cells. Particularly relevant is the plasma proteins, which are going to have a big effect on osmosis later on. As I mentioned, is that other sheets do often use different terms, such as here it uses blood pressure. OCR, as I said, are really keen on using hydrostatic pressure instead, so feel free to correct that on your diagram as well. This high pressure from the arterial end all just comes from heart contractions. One of the big differences between tissue fluid and blood plasma then is that tissue fluid does not contain plasma proteins. As I said before, they're far too large to fit through the pores between the epithelial cells in the capillary walls. So that is one of the big key differences between tissue fluid and blood plasma. The tissue fluid will then just be surrounding the cells of the tissue. Because those plasma proteins have remained in the blood, is that they do have an osmotic effect on the blood plasma and on the tissue fluid. This exerts a pressure which is called oncotic pressure, and this is going to draw some of the water back into the capillaries from the tissue fluid. Now at the arterial end, blood pressure is greater than oncotic pressure, so the overall movement of fluid is going to be out from the capillaries to the tissue fluid, which is why we do see the formation of tissue fluid at the arterial ends. Now at the venous end, so the end that will then drain into venules and into the veins, the blood pressure is much lower after it's passed through the capillaries. The osmotic pressure or oncotic pressure, as OCR again do prefer to use, that oncotic pressure is now going to be larger or greater than the hydrostatic pressure because of the plasma proteins. Because they're soluble globular proteins, they will lower the water potential within the plasma that stayed within the capillaries. And this does mean that there's now a water potential gradient moving water from the tissue fluid back into the capillaries. At the arterial end, because of the hydrostatic pressure, is that fluid will still be forced out, but at the venous end, where the hydrostatic pressure is lower, then you can see net movement of tissue fluid or of fluid back into the capillaries. This all does come down to the plasma proteins, because the dissolved small molecules can freely diffuse, so oxygen, carbon dioxide, and minerals, nutrients, they can freely diffuse back and forth. They don't have a massive effect on the water potential gradient. That is all caused by the plasma proteins that again were too large to fit out of the capillaries due to the hydrostatic pressure. What we can see in this data here is that the hydrostatic pressure is much higher at the arterial end compared to the venous end, but that the oncotic pressure is actually the same for both. Now, you can also use kilopascals as a unit for this, as well as a measure of that pressure. What that means is that at the arterial end, where hydrostatic pressure is greater than oncotic pressure, plasma moves out from the capillary to form tissue fluid, but at the venous end, where hydrostatic pressure is lower than oncotic pressure, tissue fluid will move into the capillaries. Now, those last two sentences, as well as that table of example data, is taken directly from mark schemes from questions, so you get an idea there of what kind of terminology they're looking for.